by Michael K. Skinner from Washington State University, USA, presenting on the title Environmental Toxicant Induced Epigenetic Transgenerational Inheritance of Disease Generational Toxicology. Doctor, now the stage is yours.
Thanks very much. All right. So I appreciate the invitation. I'm going to talk about basically how the environment impacts biology, from everything from domestic animals to humans. And so in the past, the environment, although it was important for just living and so forth, it wasn't really thought that the environment could, on a molecular level, change the species or its evolution. And so that's what we'll talk about. So I'll talk about, as an example of this, how environmental toxicants can induce what's called epigenetic transgenerational inheritance of disease in a whole series of different organisms, and I'll give a number of domestic animal type examples. What this is a non-genetic form of inheritance. And so that's sort of novel in and of itself. So the genetics is not involved in this inheritance. It's actually through epigenetics. And I'll describe that during the talk. And then what I'll discuss is basically generational toxicology, the fact that if we do get an exposure, it may not affect us with the direct exposure, but it may affect our great-great-grandchildren through this type of mechanism. And so, and then the problem with CRISPR and editing and sort of coming up with some sort of therapeutic molecular approach is you can do that on one or two sites, but what we find are hundreds or thousands of sites. And so essentially it really, just like with genetics and gene expression, you can do a specific genetic site to expect a specific gene expression event, but if there's hundreds or thousands of genes shifting, you don't necessarily affect the bigger picture. So unfortunately, the CRISPR is quite useful. It's a very nice experimental model sort of tool, but I don't really think it's going to get us in a place where we can fix these types of events, because these are much more genome wide. So now if in future technology would allow us to do a genome wide approach, then that's something we can consider. But that's sort of the limitation today.
Thank you. That was very interesting.
Thanks.
Thank you for answering the questions, Doctor. Once again, I thank you for being a part of this webinar and hope to associate with our future events in person.